Yeah, she's holding on inside. And Nick is back from the dead. You must have been pretty sick.
Yeah, it just didn't go away. I was sick for like 2.5, 3 weeks. Just wasn't cold. I thought.
No, I've had bad colds that just will not go away. I'm one of these terrible people who comes to work when I'm still sick, but sometimes you just, this is a cold. I feel awful, and it's just that way.
Yeah, I started getting sick like before Thanksgiving break.
That completely stinks.
I'm sure Steel will do something. Make an incision. Somewhere.
Okay, Josh, can you hear me all right? You can? Addie, can you hear me all right?
Yeah.
Good. How are you feeling, Addie?
I am okay.
You okay?
Yeah.
Josh, you feeling better or about the same?
I'm feeling a lot better, actually.
Oh, okay. Feeling a lot better. All right. Okay. Well, looks like most of us are here today. So welcome, welcome, welcome. And we're going to finish this up. I doubt this will take the whole period, in which case I have no guilt in letting you guys go a little bit early. So you're welcome for that, by the way. So we are going to talk about development and how changes during development change the form of the organism. And then we will use Monday essentially as a review session. So please come Monday with things to ask about. All right, let's have a word of prayer. Lord, thank you. It's a little warmer today, and we're very grateful. Thank you for the sun. We thank you for this last full week of school. We pray, Lord, you'd help us to focus, to buckle down, to really get ready for finals. We pray that I would write a good final, that the students would take a good final. Thank you for all the things you've taught us this semester now, Lord. Continue to change us from the inside out through the work of your Spirit. In Jesus' name we pray. Amen. So we saw, this is the example I gave you last time, that you have different cell types when it comes to these two nematodes. So I've got to do double duty here because they're on Zoom. So remember that these worms, they come in two types. There's hermaphrodites that have eggs and sperm, and there's males. And the hermaphrodites will not self-fertilize unless they have to. That's to keep diversity, essentially, to make sure that if the hermaphrodites mate with those that are just true males, they will essentially have genetic diversity in their offspring and you don't get inbreeding. So the development of the vulva is from the ectoderm, Now, I should say this, the development of the worm is what we call a mosaic development system. Mosaic development system, what is that? Mosaic development system means that when the zygote starts to divide, what that particular zygote, that so-called blastomere is going to become is written in stone once it divides. And then it will divide, and then its progeny are going to become this cell type, that cell type. You can actually map out exactly which cells in the embryo are going to become what cell in the adult. So if you take a laser and you ablate a certain cell in development, you'll still get the worm that develops, but it'll be missing those cells that develop from that particular blastomere. This is different from what you see, say, in zebrafish embryos, where you have a yolk sac and on top you get a blastodisc. If you pull out some cells from the blastodisc, the other blastodisc cells just divide and replace them. And if you take one of these cells and put it over here, or take one of these cells and stick it over here, that cell now senses the environment it's in and changes its developmental fate. That's called a regulative developmental system. And humans, at least human development, mammalian development in general is more regulative than it is mosaic. So if you get rid of the anchor cell, because the worm people know exactly which cell gives rise to the anchor cell, you don't get a vulva. The anchor cell secretes glycoproteins that signal to the overlying embryonic ectodermal cells, you're going to become vulval cells. And so they become either these primary or secondary or tertiary cells based on how close they are to the anchor cell. Right up against the anchor cell, you become primary. A little further away, you get a little less of that secreted glycoprotein, you become secondary, and if you're further away, you get a lot less of it, you're tertiary. If you're so far away, then you don't really get any of it, you're ectoderm. And we call that fuse here, I'm not sure why. They call it quaternary here, and I'm not sure why, but as you can see, these cells essentially also are primed to be able to respond to that secreted glycoprotein by virtue of this Lin39 gene. So it's not that there's a difference in the anchor cell. It's not there's a difference in the secretion of the wingless ortholog from the anchor cell. It has to do with the cells that are primed. And these cells are primed by Lin39. In this related nematode, that area of receptivity is narrower. It's narrower because the expression domain of Lin39 is smaller. So it's not the particular gene products themselves, it's the expression of those gene products. We're going to see this again and again and again. We see it in the worm. We're also going to see it in our favorite friend here, the insects. So Drosophila melanogaster development has been worked over ad infinitum, ad nauseum, ad megamesicum, basically. It's really, really well understood. So once people looked at the fruit fly, they started looking at other insects. Now in insects, you have what's called a long germ band. You have long germ band insects, and you have short germ band insects. What is the germ band? The germ band refers to this area on the ventral part of the embryo. And the ventral part of the embryo is going to give rise to the mesoderm. So the mesoderm is going to fold up and go into the the animal. And once it goes into the animal, it is going to round out and it's going to become the gut. And cells essentially from that will delaminate off and become the mesoderm. It'll form the well, no, that forms from something else. It forms muscle and things like that. And then the tube forms the actual digestive tract. The ectoderm that then comes and replaces that will become the neural ectoderm that forms the nerve cord. Because remember, insects have their nerve cord on their belly. We have ours on our back. We have a ventral gut and a dorsal nerve cord. They have a dorsal gut and a ventral nerve cord. Dorsal heart and a ventral nerve cord, sorry. Ventral heart and a dorsal nerve cord, the other way around, dorsal heart, ventral nerve cord. So this is the germ band. And when that forms, essentially, this part of the embryo is actually going to go like this. It's going to extend up like this. And this tissue here is called the amnioserosa. And what that tissue does is as the germ band extends, it essentially is going to give like an accordion. And it's going to do that. And there are mutations where the amnioserosa is too stiff and the germ band actually dives under it and it decapitates the embryo. So you have things called short gastrulation and then twisted gastrulation and folded gastrulation that actually do this to the amnioserosa. And you can see the amnioserosa here is on the dorsal side of the embryo. And it's different in these short germ band insects over here. All right, so this is a long germ band insect, Drosophila melanogaster. Now there is a gene that's expressed along the dorsal part of the embryo. It's called ZEN, Z-E-N. Then if you stain the embryo for Zen RNA, it makes a mohawk right down the dorsal part of the embryo. And if you don't have Zen, you essentially have an embryo that has two ventral sides. So you have a double ventral embryo. That doesn't survive, as you might guess. So without Zen, you can see you don't make an amnioserosa, and this dorsal ectoderm just extends all the way up to the top of the embryo, and you get kind of this double dorsal phenotype. There is another gene that's called TOLL, T-O-L-L. Now, TOLL is very interesting because it plays in insects a very unique role in dorsal ventral patterning. It's also expressed in the insect immune system. Toll-like receptors play an important role in our immune system. Toll-like receptors are on white blood cells, and they determine, well, first of all, they use them to bind to pathogen-associated molecular patterns, PAMPs. They bind the PAMPs, and that activates them. The dorsal ventral signaling in the embryo, mammalian metazoans, other higher metazoans don't have that. This, however, is used in the insects. So mutations in TOL that you see completely wipe out the ventral part of the embryo, and essentially this dorsal part replaces the ventral part. You don't get this stuff in green, the neurogenic ectoderm. You don't get the mesoderm forming. You don't get any of that. It's just all double dorsal. So Zen gives you double ventral, TOL gives you double dorsal in the fruit fly. So this is Tribolium. This is the flower beetle, the flower beetle. And if any of you worked in a bakery, occasionally you might see these in your flour if you don't store your flour properly. These are little black beetles. If you've ever, some people who have pantries where they store a lot of stuff, you'll see this usually people who live in really rural areas because getting to the store is hard. They'll buy, tons of flour. It's like going to Sam's Club and getting, the big bulk flour, that sort of thing. And they'll open the package of flour and they'll just kind of crinkle it shut like that, but maybe they don't use the flour for a while. They go in and then there are these little black beetles in it. That's Tribolium. So Tribolium is really easy to grow in the lab because you just grow it on flour. Flour is cheap. And Tribolium, as you can see, the amnioserosa takes up a much bigger part of the embryo. than it does in melanogaster. You still have some of these other features. You still have this amnion layer here, and you have some of these other features that you'd expect from the embryo, and then you have this thing called the posterior pit that marks essentially where the back of the embryo is going to be. You'll get this in the embryo and Drosophila and melanogaster, but later, because the pole cells are actually going to form here. And then when the germ band extension occurs, they're actually going to go gulp into the embryo, and they're going to migrate through that and then migrate into the ovary. Well, the ovary or the sperm as it forms, that's segment 5, the segment 5 mesoderm. And the ovary forms, and they essentially form around the germ cells. They don't need the germ cell to form, but they form around it. So you get the posterior pit here, and this is your amnion structure here, and this is some of these other structures here. And this is your dorsal side, this is your ventral side, and then this both dorsal and coming over the top. And so here's your wild type here, essentially later on, where you have your neurogenic ectoderm here, your dorsal ectoderm here, and then your mesoderm here. Same song, second verse here. You can see those 3 things here. Now, Zen RNAi, you're using an antisense Zen RNA, you're injecting it into the embryo, so you're inhibiting Zen. What does Zen do in the fruit fly? It prevents you from making dorsal structures. Without proper Zen expression, you don't get dorsal structures. So if we look at this here, notice that the amnioserosa is completely gone. Just like what you see over here. The amnioserosa is gone. You still have these basic structures, but this part is gone. Same thing over here. You keep these, but the amnioserosa is gone. So essentially, this part of the embryo that had this amnioserosa, notice what's different is where it's positioned. But the gene does the same thing, regardless of where that amnioserosa ends up being. What does Zen look like in the tribolium as opposed to the fruit fly? Really similar. You can take the tribolium Zen, put it in the fruit fly, and it works. So it's not the difference in the gene. It's where the gene's expressed. Where it's expressed determines the position of the amnioserosa. Without Zen, you lose it. What about toll? Without toll, we lose these ventral structures. Well, looky here with toll. This dorsal structure, the amnioserosa, is unaffected. Now you just have the dorsal ectoderm all across the embryo, just like you do here. The big difference is that you lose the amnioserosa. Here's where you actually expand it over here, because you've lost essentially these other tissues that were probably pushing it away. Now it expands down here to essentially take up the slack from these other tissues that you lost, and you've lost the posterior pit as well. Posterior pit in tribolium seems to be made through an interaction of these different areas. You go to those areas, you don't make a posterior pit. Notice that what the gene is doing is the same. The difference is the location of the structures. That seems to be the reason why you get these differences in form. It's not due to the gene products themselves. It's actually due to where they're expressed and the tissues they elaborate where they're expressed. That's what gives you the difference in form. This is work from Sean Carroll's lab. Sean Carroll is at the University of Wisconsin at Madison. Sean Carroll's been interested for a very long time in form in the fruit fly. What drives its form relative to related species? So here we have dipterans. Many of these are very closely related to Drosophila melanogaster. The wing in these flies has a very similar shape. What's different is the coloration. What determines these differences in coloration? That's what Sean Carroll and his postdoc, Big and Prudhomme, thought to discover. And this was their paper from 2007. And Carroll has essentially extended this to other insect species since this paper. But this paper, I think, illustrated some of the really main points here. Other things that you see that are different, perhaps, in these wings, take a look at the vein pattern in these wings. So these vein patterns, as you can see, are pretty nicely reproduced in these. There's some slight differences here in position of veins, but you have essentially veins that are at the top of the wing, another vein towards the top of the wing, kind of a middling vein, a cross vein here, another vein, and then a more posterior vein here with another cross vein between. So this is called L1, L2, L3, L4, L5. And then these are called radial cross veins. This is called R1 and R2. Ethan Beer at UC San Diego has done extensive work on what makes a vein, essentially in a wing of the fruit fly. He's discovered it's due to a gene product called rhomboid. Whenever you express rhomboid, you get a vein. Ethan Berry showed this over and over and over again. If you express rhomboid throughout the wing, you get a blistered wing. Why? Because now the whole wing is vein. If you prevent rhomboid expression, you prevent the formation of various types of veins. So rhomboid seems to be associated with vein production. Ethan Beer has now extended that study to different types of insect wings. So lace wings, for example. Lace wings have tons and tons and tons and tons of veins. Beer has shown in the lace wing, wherever those veins are, rhomboid is expressed. And then he's taken it to beetles as well. So the activity of rhomboid is absolutely conserved throughout the insect group, essentially. Rhomboid makes veins. The difference is where rhomboid is expressed. And if you look at the promoters of rhomboid, in those different insects, they have different enhancers that are associated with the gene, and that changes its expression pattern in the wing. That's how you get the different venation patterns in the wing and what have you. So we can explain these different venation patterns. What about the color? Turns out the color is due to the expression of a gene called yellow. Yellow. Now here's the part about this that's confusing. In Drosophila genetics, the name of the gene is named after the mutation and its phenotype. In fact, I think when you took genetics from me, we had yellow flies in our lab. Apricot, that's the eye, yeah. And then there were the yellow, the bodies were not very dark, yeah. So the flies, instead of having a dark body, they have a pale colored body that's got a yellowish tinge to it. That's why the mutation, the mutation defines a gene. The mutation was called yellow, and that defines a gene. Turns out yellow, the gene product, turns on the deposition of melanin in the fly, in the insect cuticle. So wherever yellow is expressed, you get dark melanin that is deposited. Turns out that these spots on the wing are due to yellow expression. Now, melanogaster does not have colored wings. There's no yellow expressed in its wings. However, if you start taking the yellow gene and you express it using these different types of promoters and enhancers, which you can do using these UAS strains of flies and using the GAL4 system, you can now get wings that essentially will express different types of patterns of color across the wing. So this is due to the enhancer of a gene called SPALT. Spalt is used in several different things in the fly, but one of the things it does is it helps form the anterior compartment of the wing of the fly. So spalt is expressed in the swath across the front of the wing like that. If you express the yellow gene in the wing using the spalt enhancer, you get a wing that's that color. Remind you of something? This is a species of dipteran that has this particular color. Sean Carroll was able to completely recapitulate it in Drosophila melanogaster. If you go to the yellow gene in this species, turns out the yellow gene has an enhancer in its promoter, and Carroll has shown this, that expresses yellow in the gene in precisely that pattern. That same enhancer is found in the spalt gene. That enhancer simply got co-opted by the yellow gene. And then we have this other enhancer that is able to drive expression, not just in the cross veins, but in kind of a little leopard pattern along veins here. This particular gene, when the yellow gene was driven by the enhancer of this gene, it gave this particular pattern right here. Well, this looks a whole lot like that particular fly up there, doesn't it? And if you examine the yellow gene, it's got an enhancer a whole lot like the one that was engineered by Prudhomme and Carroll. So again, when you compare the yellow gene throughout these dipterans, the yellow gene varies extremely little. It's very heavily conserved. And you can swap yellow genes from different dipterans and put them in melanogaster and it works just fine. The difference is where it's expressed. That's what explains essentially the diversity of color in the wings. So the yellow locus essentially has enhancers that will allow it, in melanogaster at least, to be expressed largely in the body and in the legs, but not in the wings. If you now take this sort of spot enhancer, as it's called, or a blade enhancer, you can essentially then put color into the wings. And then in these flies that have these enhancers, if you mutationally inactivate them, you can remove that color as well. There are also enhancers for the body, for the dark spot down here, and then also for the stripes. If you mutationally inactivate those enhancers, you will lose color either in the tip of the body or in the stripes of the fly like that. And this is shown essentially like this. And you can see essentially the normal coloration. But then if you start to do away with this various coloration, you start to get patterns like this. Now, it turns out that Drosophila willistoni, this is a species we've talked about. Drosophila willistoni doesn't have this dark spot at the tip. If you look at its yellow gene, it does not have that body enhancer that binds the abdominal B gene. The abdominal B gene is expressed towards the end of the body. There's an abdominal B binding enhancer in the yellow gene promoter of Drosophila melanogaster. It has a dark spot here. Willistoni doesn't have that. But Willistoni still has stripes of melanin because it has that stripe enhancer. This species, Drosophila kikawai is a Hawaiian species. This Hawaiian species is able to hide amongst some of the tender blades of tall grass because it's not dark. It lacks both the body and the stripe enhancer. And if you take melanogaster and get rid of both of these enhancers, it now has a body that looks a lot like Drosophila kikawai. And if you look at the yellow gene in Drosophila kikawai, its yellow gene has neither of those two enhancers. Does that make sense to all of you? Now that's coloration. What about the actual form of the body? Well, once again, in Drosophila melanogaster, it's very well understood that there are these genes called homeotic genes that determine segmental identity. Homeotic genes form a gene cluster. Here's the kicker about this gene cluster. They're arranged on the chromosome according to where they're expressed in the body. So this is expressed in the front, then next most front, then front middle, then middle back, kind of all the way toward the back and all the way toward the back. Furthermore, homeotic gene clusters, no matter where you look at the mammals, insects, even in starfish and coral, this is absolutely conserved. Absolutely conserved. So in melanogaster, there's a gene that is called ultrabithorax, UBX. Ultrabithorax is made in the third thoracic segment, and it prevents the formation of a wing. Instead, you get those little nubby organs that are balancing organs. They're called halteres. If you pull the halteres off a fly and ask it to fly, it starts flying upside down. All this bit, yeah, pretty cruel, but hey, that's life. So it's a balancing organ. Ed Lewis at Caltech in the 60s and 70s showed that mutations in ultrabithorax transform the haltere to a wing. So without ultrabithorax, you express the wing genes in the thorax. When you look at ultrabithorax in lepidopterans that have two sets of wings on the second and third thoracic segment, you find they have ultrabithorax, but guess what? The genes for wing formation are not repressed by ultrabithorax. They've lost the ultrabithorax regulatory regions. That's why they make wings on the third thoracic segment. So dipterans in general, their wing promoting genes are repressed by ultrabithorax. In lepidopterans, that's not the case. So is the change in ultrabithorax? No. The change is in the target genes and what turns them on and what turns them off. And so Sean Carroll has done quite a bit of work on the lepidopterans. So he expressed the fruit fly ultrabithorax in the lepidopteran. It took him a long time to figure out how to do that. If you do that, you still get wings. Then he used RNAi to get rid of ultrabithorax in the lepidopteran. Can you guess what happened? These abdominal segments started to sprout wings. So the homeotic genes are saying this is abdomen. Abdomen doesn't make wings. They don't promote the formation of the wings, they tell the body where to put the wings. So the change in form has to do with how the downstream genes are regulated, not the actual genes themselves. And the same seems to go for hymenopterans that have a forewing and a hind wing, Or for example, with beetles that use the forewing is usually a wing cover, an elytra. The back wing is the wing that they use for flying and what have you. But that seems to be the case. And Steve Cohen at Embl in Heidelberg, he's done experiments showing that the homeotic genes in arthropods are largely due to repress wing formation. They're not for putting wings on, they're essentially for regulating where wings don't appear. All right. And then when we get to vertebrates, do we see a really different theme? The answer is no. So this is a stickleback. If you've done any fishing in any of the streams in Michigan, you may have come across sticklebacks, not very big. Some people, if they catch a stickleback, they'll actually use it for bait because the bigger fish, the trout, sometimes will like stickleback for bait. So you just take a little stickleback and you put it on the hook and you just dangle it. And the brook trout really like sticklebacks. And brook trout, of course, are very good eating. Okay, yep, Sam, we're on the same page with brook trout at least. So the stickleback, it turns out, has two different forms. There's what's called a benthic and a pelagic form. A benthic and a pelagic form. Benthic form tends to live deep in lakes. Pelagic form tends to live higher up in the lake. The pelagic form has these rather dangerous looking pectoral spines. I think those spines might be used for. Huh? Defense, yeah. Pretty hard to get your mouth around a nice tasty meal if it's got those spines. It's got spines essentially. You're going to get a mouthful of spines. Now, the benthic form does not make the spines because deep where it lives, there is a predatory copepod and it grabs the fish on the spines. By grabbing them on the spines, it then can pick it apart and eat it. So the spines are a detriment, actually, to the benthic form. But the pelagic form, where you've got bigger fish that want to eat it, the spines essentially are a benefit. All right, so what essentially puts those spines on the pelvic girdle? Turns out there's a gene called PITX1. PITX1, as you might guess from its name, is involved with lots of things. One of them is making the pituitary. It also helps make the face, but it also helps make limbs. So PITX1, as it turns out, is deeply involved in the formation of these spines. And when you investigate PITX1 expression in these two forms, something interesting falls out. So there was a paper, not the paper right here. So there was a paper essentially where the Kingsley lab at Stanford essentially looked at crosses between the benthic and the pelagic form. And using these crosses, they're able to show through mapping the areas in the genome that were responsible for the presence or absence of spines, that the PITX1 locus essentially was responsible for the formation of the spine. Now, it turns out that the benthic form, pelagic form, not only has a spine, but it also has head armor, and it has armor over here to keep the animal from getting chewed on, essentially from bigger fish. As you can see, the head structures, the sensory structures on the head, also depend essentially on PITX1. And if you cross these animals, essentially, they don't have the spine, you still get the sensory organs on the head. So the sensory organs on the head are not affected, even though they're influenced by PITX1. It's the tail spine, the pelvic spine, that seems to be the bugaboo, if you will. So here are some, this is called an in situ hybridization using PITX1 messenger RNA in the tissue. These are stickleback embryos that have been hybridized with the RNA, essentially, of PITX1. Go through this with me. So here's the head, and here's the tail. And here is the marine, and here is the benthic version. So the benthic version lives high in the water column. The marine one tends to live low in the water column. So here you can see you have these sensory organs here. That's called the nasal placode, and is nasal placode. That's going to help make the nose. The T over here, that's the formation of the thyroid. Is the thyroid. Then as we get to the back here, we can see that we have these elements of the lateral line here. Pretty much it's all good. A little stronger expression here than we do here, but you still have these organs forming. So the difference in the head and the lateral line, not very big. Now when we get to the tail and the pelvic girdle, things get kind of interesting. In situ, sometimes you need dark really to see them with their color. All right, so as we go around the pelvic girdle here, we look at the pelvic girdle. Do you see any PITX1 expression? No. When you look at the marine form that has the spine, now you see PITX1 expressed. So it seems to be PITX1 on, you get the spine. PITX1 off in the pelvic girdle, no spine. So when you look at the PITX1 mutation from the marine and the freshwater sorts of animals, you see something. You have enhancers in the gene. So here's the gene product itself. You have the so-called thymus, hind limb, and neuromast enhancers. In the marine version, all these three enhancers are intact, and you make these spines. In the freshwater version, the hind limb enhancer is not present. The other two are, but the hind limb one isn't. So you make your sensory organs, you make your thymus, but you don't make the spines. Now, this is the mouse here. With the mouse here, we have essentially taken the PITX1 gene and we've gotten rid of it. And we get something that looks really interesting. Look at this and tell me what you see here. Much shorter. Do they look more like hands and feet? In fact, they do. In fact, those bones have been transformed into forelimb bones. So these mice obviously die very early during embryogenesis, but the hind limbs have been transformed into forelimbs. And in our bodies, PITX1 is expressed in the forelimbs, not in the hind limbs. And there are mutations in PITX1 that are pretty subtle, but they cause hind limb defects. They cause patellar defects and things like that, and other types of things like that. But this gene is still present in the stickleback. Those two versions of the stickleback, the pelagic and the benthic, they have the same PITX1 gene. The difference in the form is how it's expressed. So here again, we just put this up here essentially just to reinforce the point. We saw this in insects. We see this now when we look at vertebrates. And there's other work that you can include in this, but this theme seems to really repeat itself over and over again. Changes in form seem to be not so much due to differences in the actual sequence of genes per se. That's not to say sequences of genes aren't important. They are. So for example, in the brine shrimp essentially have leg appendages essentially on every segment. If you look at the ultrabithorax gene in the brine shrimp, it is missing a little piece of the DNA binding domain, so it doesn't bind and repress limb formation on all of the segments. So yes, absolutely, the coding region can be important, but what seems to be more important is the expression of genes, changes in the expression of genes, not so much the sequence of the genes themselves. And by changing where genes get expressed early in development, you can alter the form of the organism in pretty profound ways sometimes. Ways that actually give you two different populations that occupy two different parts of the water columns, and now in nature effectively never see each other. And those two are on the way, it seems. Well, some people would argue that they are separate species, because it doesn't matter if you can get them to mate in the laboratory. The fact is, in the wild, they never encounter each other. So they have experienced reproductive isolation. All right, so that essentially is that's Evo-Devo essentially. And I think it drives home a point that's pretty important when it comes to the themes essentially that we've talked about in this class. Now, one of you emailed me and said, well, Dr. B, you gave us these options, but you didn't tell us what you think. Yeah, nice try. I'm not going to tell you what I think. I want you to figure out what you think without me contaminating your thinking. So I think to be a Christian in today's very secular world requires a certain amount of, excuse the expression, tough-mindedness. It requires people to think and think deeply about complicated issues. These are not simple issues, as we've looked at. So I would like you to do that, and I will tell you right now, one of your questions on the final is going to be, what do you think about all this? So do you have to read me the riot act within 2 hours of taking a 50 to 60 to 70 question final? No. If you're unresolved on certain things, that's fine. But I would hope you've come to some conclusions, and I'd like you to be able to articulate those in some kind of essay form in a paragraph or two, essentially. Again, you don't need to read me the riot act on it, but I would hope that you'll be able to come to some sort of resolution. Are there any questions about anything we've talked about? Our Zoomers, do you have any questions About anything we've talked about? I see Josh shaking his head. Addie, your camera's not on, so I can't see you shaking your head.
No, I'm good.
You're good? Okay. Well, if you're all good, then I will let you go early and feel no guilt whatsoever. So I will see you guys on Monday. Please come with questions for me to hopefully try and answer. Please don't come and ask me, so what's on the test? Because I will not have written it by then. Let me just tell you, I don't know. You might notice it's a tad warmer in here. Charlie Regan was in here essentially.